call the code. Push 5 milligrams epi. Clear the area. Charging. Clear. We're losing him. Time of death, 11:55.
Good morning, everyone. In today's seminar, we are going to be discussing aspects of T-cell alloreactivity and how they relate to transplant rejection, which is what the patient in the video experienced for a few months prior to his death. We will briefly cover the basics of T-cell development, MHC-TCR interactions, and peptide specificity and how they all lead to T-cell alloreactivity. I'm assuming everyone has done their readings and is able to discuss this topic, right? Remember, you will be graded on this. So just to overview, T-cell alloreactivity is the ability of T-cells to recognize foreign MHC complexes that have not been recognized during thymic development, typically from a donor in the case of organ transplant. Since these host alloreactivity T-cells are able to recognize peptide allogenic MHC complexes, the adaptive immune system is able to mount a defense against the donor MHC expressing cells, leading to rejection of the donor organ. In the case of graft versus host disease, alloreactivity of donor T cells can lead to recipient tissues being attacked. We will go into more details in our discussion later. Altogether, alloreactivity has huge implications in immunology, especially in the clinical field. Before today's seminar, I will show you this video prepared by my colleague, Dr. Zhu.
What is up, guys? I'm Dr. Greghi, here with my good friend, Dr. Zhu.
Hi, guys.
Today we'll be talking about T-cell recognition and T-cell development in the thymus. Let's get into it.
During the development for T-cell in thymus, each T-cell's receptor, which is TCR, undergoes a VDJ rearrangement. The TCRs consist of alpha and beta chains here. The alpha chain is produced by only VG recombination, and the beta chain is produced by VDJ recombination. The random recombination of gene fragments provides a very large diversity of the production of TCR, and the diversity of TCR is the basis for the recombination of many different antigens of the T cells.
So in fact, it's actually not quite that simple, as there's still a few things we must do in the thymus before itty-bitty T cells become fully developed. All right, so in the beginning, we must do positive selection, where we have these T cells that show both, as you can see here, CD4 and CD8 receptors. And what these will do is they'll be tested against MHC molecules, and we'll determine which one has the higher binding affinity. At this point, it becomes a battle to the death between the two receptors. And whichever one has the higher affinity towards MHC will survive, and thus the T cell will become a helper or cytotoxic T cell. So now that our T cell is capable of forming a complex, we must make sure that our T cell is not going to contribute towards autoimmunity, okay? So to do this, we will go through negative selection inside the thymus, where our T cell here, we're going to test the TCR receptor against self-antigen. Any of these T cells that have too high of affinity or are binding to multiple self-antigen will be undergoing apoptosis to remove them from the T cell population.
These T cells, which develop in our bodies and pass through positive and negative selections within our thymus, are self-tolerant in our bodies and can also respond to the foreign antigens present by our cells' MHC molecules. They will respond for the conventional antigen recognition, which is one of the foundation of the immune response.
Jenny, can you explain the concept of MHC polymorphism and proposed models and how they all relate to alloreactivity?
Oh, I'd be glad to. Due to MHC polymorphism, there's hundreds of MHC alleles in human population. However, each individual can only express up to 6 MHC1 and 6 MHC2 alleles. Therefore, although host T cells can recognize self-MHC complex, there is no selection of host T cells against foreign MHC variations. Since there's conserved regions on the MHC across all humans, alloreactivity of TCR could be the result of its inherent affinity for foreign MHC molecules, without the need for prior exposure or selection. In other words, even though they're trained to recognize self-MHC, they can sometimes recognize non-self MHC molecules, like those from a solid organ donor. Now, as mentioned in the YouTube, in conventional recognition, T cells are selected in the thymus to recognize self-MHC molecules bound to self or foreign peptides. In this interaction, the TCR typically engages both the peptide and the MHC molecule in a specific manner, allowing the T cell to distinguish self from non-self without triggering an immune response. Whereas for allorecognition, it is proposed that TCR often binds to the allogenic MHC molecule with or without strong dependence on the bound peptide. For example, the high determinant density model suggests that TCRs may react primarily with the MHC itself, whereas the outer self model suggests that TCR interact with the bound peptide alone. And these unique interactions are what distinguish allorecognition from conventional recognition. However, there's no conclusive evidence to either model because every TCR peptide MHC structure we discovered so far involves some degree of TCR interaction with both peptide and MHC molecules. Current evidence suggests that it could be a mixed model where TCRs likely interact with both MHC and peptide in various degrees, depending on the context, rather than a strictly peptide-independent or peptide-dominant approach.
Thank you, Jenny, for this great and thorough explanation. Now, Mesai, do you want to introduce us to peptide specificity, polyspecificity, and how they relate to some clinical aspects?
Thank you for giving me the opportunity to speak. So an interesting fact is that many alloreactive TCRs recognize specific peptides presented by the foreign MHC, resulting in targeted peptide-specific immune responses. So this allows for peptide specificity, which is an interaction between the TCR and peptide MHC complexes determined by the similarity between the donor and the recipient MHC alleles. So on the other hand, right, we have with greater MHC disparity, TCRs may respond to a broader array of MHC complexes, even if they do not share similar structures. So this is called peptide degeneracy, right? So where there's less interaction between the peptide complex, and this currently is evidence to show that conventional TCRs are more degenerate, whereas alloreactive TCRs are more specific. So this is where polyspecificity comes into play. Polyspecificity refers to the ability for a single TCR to recognize multiple unique peptide MHC ligands while maintaining specificity for one which is more prominently shown in alloreactive TCRs than conventional. So in the context of transplant rejection, polyspecificity allows for TCRs to recognize and react to several peptides from a donor's MHC, potentially triggering a more extensive immune response, right, against the transplanted organ. So then bringing all this together, the peptide specificity, polyspecificity, bringing all of this together, this allows for transplant rejection, which is when an alloreactive TCR recognizes the donor MHC and they mistakenly trigger a response against the transplanted tissue, leading to a rejection, right? So immunosuppressive drugs help reduce the likelihood of this rejection by dampening the immune system. But as we saw in the video with the person on the gurney, this increases susceptibility to infection, while other TCR recognition mechanisms offer flexibility and protection against diverse pathogens. They also pose challenges in transplants by recognizing and attacking foreign tissues.
Thank you so much for the passionate speech, Mesai. Now, Tim, do you want to go over some specific implications on this topic?
Yes, of course. The modern understanding of T-cell interactions has revolutionized the field of solid transplants, allowing for many people to live a healthy life with the aid of a foreign organ. T-cell research has allowed for solid transplant organs to become more widely accessible. Even with only one match out of 6 MHC alleles, this is feasible due to the development of immunosuppressants. As of currently, the leading factor towards transplant success rate is ABO factors, or in other words, blood type compatibility. However, when it comes to bone marrow donation, the story is very different. The goal in hematopoietic stem cell transplants is to enable donor immune cells, including T cells, to mount an attack after recognizing foreign peptide recipient MHC. Thus, this requires the MHC genetic data to be as similar as possible to prevent autoimmunity or the failure to recognize recipient MHCs.
Thank you guys for the awesome participation. I'm glad almost everyone participated in this discussion. So altogether, Understanding the mechanisms of T-cell development and alloreactivity is vital for advancing transplantation science, improving graft survival, and developing therapies to modulate immune responses. Insights into TCR degeneracy and alloreactivity can lead to novel strategies in immunotherapy, such as engineered T cells that can be made less reactive to non-self MHC. Further research could focus on identifying specific TCRs involved in alloreactivity and developing targeted approaches to mitigate adverse transplant outcomes, ultimately enhancing patient care and outcomes in transplantation and autoimmunity. So class, if there's one thing I would like to take away from this discussion, is to make sure you are kind and caring to your relatives, as you'll never know when you'll be needing their hematopoietic stem cells. That's it, guys. You're all free to leave.